Have you ever thought about what's happening inside you right this second? It's not quiet in there. There's this incredible, silent conversation going on between trillions of tiny machines we call proteins. They're sending signals that control literally everything. And today, we're going to learn how to listen in.
So here's the big mystery that's had scientists scratching their heads for years. It's this thing called allostery. How can you poke a protein in one spot, maybe with a drug molecule, and cause something super specific to happen way over on the other side? I mean, on a molecular scale, that's a huge distance. It's kind of like pressing a doorbell in New York and having a specific light turn on in Los Angeles. How on earth does that signal travel?
You know, the old answer was always just shape. You push it here, and so it bends over there. Simple mechanics. But it turns out that's not the whole picture, not even close. The new idea, the one we're diving into, is that it's less about moving parts and a whole lot more about acoustics. We've got to stop picturing proteins as little Lego blocks and start thinking of them as what they really are: incredibly complex musical instruments, each with its own unique, constant hum.
All right, so to really get this new idea, we've got to first talk about why the old models, as good as they were, just couldn't explain everything. They were missing a key ingredient. They were missing the music.
Okay, so the old view basically saw proteins as these clunky switches. You flip one part, and another part just clicks into place. A big, obvious change in shape. But here's the weird part. Scientists kept finding examples where this long-distance communication was happening with almost no visible change in the protein's shape at all. The message got sent, but the protein barely seemed to move. And that, right there, is the new reality. It's a puzzle that needed a whole new way of thinking.
And that new way of thinking, it's a total paradigm shift. It comes from seeing proteins not as static things, but as these dynamic, constantly vibrating, humming networks of energy. And this is where the music starts to get really interesting. Scientists can now use this really cool tool called an elastic network model. Basically, it lets them ignore all the tiny, complicated details and see the protein for what it is: a web of points connected by springs. Think of it like seeing the protein's skeleton. It's a brilliant way to reveal the big picture of how the whole structure wants to move and flex as a single unit.
So when you analyze that network, you find something amazing. Every single protein has these things called low-frequency modes. Now, forget the technical term for a sec. Let's go back to that cello or guitar. These modes are the deep, resonant notes. It's not the sound of plucking just one string. It's that fundamental hum where the entire body of the instrument vibrates together. For a protein, these are its most important collective wobbles, the motions that are essential to its job.
And hey, this isn't just a nice little metaphor. The science is solid. As researchers like Wodak have pointed out, there's tons of evidence showing that these collective hums, these so-called soft modes, are directly linked to how proteins actually work. They are the absolute key to understanding that long-distance communication because they involve the entire system acting in perfect concert. So, if proteins have this natural, built-in hum, what happens when something like a drug molecule shows up? Well, it doesn't just push it. It does something much more elegant. It retunes it. Okay, so check this out. Here's how it works. Step 1, a protein is just hanging out, humming along with its natural vibrations. Step 2, a molecule comes and binds to one spot. Now think of this like gently placing your finger on the body of a guitar. You're not moving the guitar, you're just making one tiny spot a little stiffer. And what happens? That one small touch instantly changes the resonance of the entire instrument. The fundamental hum shifts, and that new vibration, that new music, changes how the protein works at a totally different site. That, my friends, is mode shifting. So let's just boil it all down to one core idea. Allosteric regulation, that mysterious action at a distance, is all about redistributing a protein's vibrational energy. We're not pushing puzzle pieces around anymore. We are literally retuning the symphony of the cell. Now, this is a really profound way to think about biology for sure. But it's not just a cool theory. The question is, what can we actually do with this knowledge? Well, if we understand the music, we can start to find the master switches, the exact spots on any protein that have the most control over its tune. And this brings us to an incredibly powerful new tool called spectral leverage. It's essentially a calculation. For any point on a protein's surface, we can now calculate exactly how much vibrational influence it has. In other words, if we press down right here, how much does it change the protein's overall hum? It's like a map that shows us all the hidden control knobs.
And this is where it gets really game-changing. Think about what this means. We don't have to spend years doing trial and error in a wet lab anymore. We can now just take the 3D structure of a protein, which we often already have, run it through a computer, analyze its geometry, and predict where the hidden control sites are. We can find the best places for a drug to bind before we even step into the lab.
And does it actually work? Oh, you better believe it. The data is just beautiful. When they ran these calculations, they found that the spots we already knew were important allosteric sites, the ones confirmed through decades of research, they had sky-high spectral leverage scores. And just random spots on the surface? Almost nothing. It's the proof in the pudding. The master switches are exactly where the math says they should be.
So, what's the big takeaway here? Why does this all matter so much? Well, the implications are just massive. It means we can find new places to target with drugs, and way faster. We can design smarter medicines that don't just shut a protein off, but that carefully retune it to work better. We can finally understand why some tiny genetic mutations cause devastating diseases. It's because they mess up the protein's hum. And for fields like synthetic biology, well, it's a whole new set of rules for building biological machines from scratch.
So let's just zoom out for a second and appreciate how big a deal this really is. This isn't just a neat trick for studying proteins. What we're talking about is uncovering a universal language that complex systems use to communicate.
By thinking in terms of things like vibrations, frequencies, and networks, we're putting biology on the same playing field as physics and engineering. The same powerful ideas that a physicist uses to understand why a bridge resonates in the wind, or that an engineer uses to design a circuit board, we can now use those same ideas to understand life itself. The messy, complicated world of biology is starting to speak the beautiful, simple language of physics.
It all comes down to this: that mysterious process inside a cell, the flow of heat through metal, the way a signal travels down a wire. They can all be described using the same fundamental principles of networks and vibrations. We're finding out that information moves through the universe in remarkably consistent ways, whether it's in a living thing or something we built ourselves. And that leaves us with a pretty mind-blowing final thought. For so long, we've tried to understand life by taking it apart piece by piece. But now, we're finally learning how to read its music. So, the real question is, once we get good at not just reading that music, but at writing it, at composing brand new symphonies inside our own cells, what incredible things are we going to ask them to do next?